So what, as Christian said, I mean, this will take you into a journey of evo-devo while you're focusing on population genetics. Not for me, but I don't know. If you don't fall asleep, it's maybe better for the picture is better, yeah? So, and as I see it, I mean, other people before me have coined this, what's all about evolution. I think there's two major aspects, and we can summarize them as pattern and process. And I think what you're mostly interested in here are the processes of evolution. And when you go deep in evolution, so deep in time, I mean, then we don't have access anymore to population sizes and all these other aspects that you need to understand the processes. Instead, we see the patterns of things that have been fixed, and that's what we are looking at, And so I try to give you a little bit of insight of what we have been doing using this model organism. Now I have to learn how this works. Yes. So I would like to start with this picture with many different animals and putting a bold question on top of it, like how did complex multicellular organisms evolve? And what's interesting here is that they all look extremely different in terms of their body plans. But of course, these are all adult forms, and they're at the end of their developmental becoming. So every organism starts with a fertilized egg, and which in the beginning of their development is often very similar, and then differences occur with developmental time, and then they get split into different species, and in the end you have very differently looking body plans. What we try to address this question about is these major animals here. So on the right you see Nematostella bectensis, the sea anemone that I established like 25 years ago, but the other, they belong to the same class as these box jellyfish here on the left. So they belong to the cnidarians. What are the cnidarians? I mean, the cnidarians are, morphologically speaking, quite simple. So you have, at least taking Hydra with the freshwater polyp as an example, it's rather simple. So it has one apparent body axis. Let's see if I manage this. Oh, I make it. Here, one apparent body axis, which we call the oral aboral body axis. So it's radial symmetric around this ring of tentacles. Consists of only two cell layers of epithelial cells. Has no organs. Has a diffuse nervous system of neurons in both layers, and a number of other small cell types like neurons and gland cells and cnidocytes or nematocytes, as well as the interstitial stem cells, which give rise to these different cell types, actually all except for the epithelial cells. So in total, just by morphological criterions, we could just differentiate maybe something like 12 morphologically, wow, this is difficult, cell types here. So really simple, and you would wonder, what does it take to make such a simple organism in terms of genetic repertoire? You would think, okay, that doesn't take much. But actually, when we sequenced the genome, this was the first non-bilaterian genome at the time, and the big surprise, at least at the time, was actually this transcriptomes and the genomes of the sea anemone were surprisingly complex. So now we know, now that we have also resequenced the genome and have now chromosome scale genomes, that Nematostella has about 25,000 protein coding genes. The gene repertoire is really comparable to humans, even on the individual gene level. So if you look at on conserved genes where you can align the protein levels, then 80% of the ancestral introns positions are conserved to the nucleotide level, even though they have been separated for about 600 million years. And so there's a lot of gene repertoire, and that's, for some time ago we thought, okay, why are they genetically so complex? Maybe they don't have very simple cis-regulation, and that makes the difference in their complexity. But then we found that actually their promoter and enhancer epigenetic landscape is using the same histone modification marking enhancers and promoters, and we have distal enhancers. So just like, at least qualitatively speaking, just like other bilaterians. And I don't have the data here with me, but it's actually, if we do the comparison, how many promoters and enhancers we have, the ratio, it's very similar to Drosophila. In vertebrates, we have more enhancers, so there might be actually, like in quantitative level, there may be an increase here, but at least with compared with Drosophila and other bilaterians, it's quite comparable. So it remains a bit of a paradox. Why they have such a complex genome, but morphologically make so little out of this? I don't have an answer to this today, but I want to focus on a couple of subjects that we work on. So first, to put you on the map. So as you probably know, cnidarians are this non-bilaterian group of animals which are the sister group to all the bilaterians. What is not shown here is there are 2 more or 3 more basically branching metazoans, like the sponges, ctenophores, and placozoans. But what's important here is that they're the sister group to the bilaterians, and I think as such they are very informative as an outgroup to understand the evolution of some key bilaterian traits, which, as I listed here, I see the central nervous system, which most of these bilaterian phyla have. They have 2 body axes, so the bilaterality, and they have 3 germ layers, while, as I said before, cnidarians consist of only 2 cell layers, which have been termed endoderm and ectoderm. So for a long time, it was thought that the mesoderm has evolved after the transition, after the split, after the cnidarians have split off. And given that, like 31 out of the 35 described animal phyla are comprised in the bilaterian, so there's an enormous radiation of body plans that we, went along with the onset of bilaterians. So it's of course tempting to speculate here that the evolution of these key features might have played an important role in playing around with a variety of different body plans. And we worked on all of this, but today I will focus on one aspect, that is kind of the evolution of germ layers, in particular the mesoderm. Yeah, so I will, and what also the consequences are from this in the other aspects. But before I go into this, I would like to first, this is a slide that I was throwing in the last moment, talk about the genome for a moment. Now that we have a chromosome scale genome, so when we do, they have 15 chromosomes, and then, for instance, here, if you have a related species called Scolanthus callimorphus, which is from the same family, but still about 170 million years separation. For a slow evolving cnidarian, that's very close, but you see that they, and each dot here, this is an Oxford plot, each dot is an orthologous gene. So you can clearly see that all the 15 chromosomes are conserved between the 2 species. And most of the orthologous genes are all on the same homologous chromosome. But what's also interesting is that within the chromosome, the genes get scrambled a lot, So, but they rarely leave the chromosome as an ancestral linkage group. But if you do the comparison with other organisms, for instance, this is Drosophila. The comparison between Nematostella and Drosophila, so has been completely scrambled. So we don't see any ancestral linkage groups anymore in this comparison. But if we compare it, for instance, with Branchiosoma, so Amphioxus, then we find quite a lot of ancestral linkage groups conserved, which may reflect directly the chromosomes or partial parts of the chromosomes. And similar, for instance, even in vertebrates, which underwent 2 rounds of whole genome duplications, then the picture looks a bit more complex, but still you see these blocks of macrosynthetic conservation. And we can use this information by pairwise comparisons between all kinds of animals to kind of reconstruct the evolution of these ancestral linkage groups, which you can take as a proxy for chromosomes, and bring them down to to the metazone ancestor and then bilaterian ancestor and so on. I don't want to go into any of these details, but it's quite interesting to reconstruct the history of the chromosomes, which really dates back more than 600 million years in many organisms. But as I said, I mean, Drosophila has gone a bit wild, but you still see these traces in many other organisms. Okay, yeah, and this is also, again, you can reconstruct this to a level where you can actually predict which, if you have here this ancestor, just taking one example, the ancestral metazone ancestor, the predicted chromosome here, number 1, which has then split into 2 at the ancestor of the cnidarians, and into 3 in the ancestor of bilaterians, as an example. So good, but I would like to come to these questions of how these key traits have evolved. For instance, the germ layers. As I mentioned before, they consist of only 2 cell layers, which typically have been called ectoderm and endoderm. So for a long time, everybody thought, okay, the mesoderm is what's lacking, and this was actually the topic of my postdoc. And when I was thinking about a project for my postdoc, it was actually Dieter Tautz, the supervisor of Christian, whom I asked, is that a reasonable question to ask, and at least he didn't advise me against. So he said, well, let's just go for it. It's crazy, but try to find it. So I wanted to find the origin or the traces of the mesoderm. And the approach was that I thought, okay, let's look for genes that are conserved in mesoderm formation in bilaterians like Drosophila, so protostomes, and vertebrates. So if that is conserved between mostly transcription factors which play an important role in the development of mesoderm in both Drosophila and vertebrates, then I took the assumption that, okay, maybe then we find these genes also in cnidarians, the sister group. Remember that the genomes were not available, so we couldn't just look it up. We had to clone the genes by degenerate PCR. To take a long story short, we cloned a lot of, in the meantime, quite a lot of mesodermal genes and looked at the expression pattern. And now we have actually, a couple of years ago, we looked also at endodermal marker genes from bilaterians because we always took it for granted that, of course, this is the endoderm. But when we found that most of the mesodermal genes are expressed in here, so maybe we thought, okay, is that the mesendoderm, where all the cells have like a dual identity, or is there some sort of segregation? And it turned out that many of these genes are really, of the endodermal marker genes from bilaterians, we find really expressed in what gives rise to the pharynx. This is the pharynx region of still a gustral stage, but it will develop into a pharynx with derivatives like digestive gland cells, but also insulin secreting cells, which are derivatives of the endoderm tissue, typically in bilaterians, while this inner layer that we used to call endoderm actually expresses all of the mesodermal characteristic genes. So what we think now is it's still diploblastic in the sense that it has 2 cell layers, but it has already segregated into 3 germ layer identities. And then this explains it also much better than you cannot suddenly come up with a completely new cell layer with all the genes involved in there. You basically express it first already in some precursor, which in this case, the scenario would be all you need to do from such an ancestor, if you take that for a moment as an ancestor, you would have to also split this tissue in terms of layers from the rest of the others, so have them in 3 layers then. Okay, so this suggested that there is a topology of 3 different germ layer identities, which I consider now homologous to the 3 germ layers of bilaterians. And if you look very early in development, like here, for instance, the late blastolar stage, we have gene expression which would be marked and would be look like that. So that some early mesodermal genes would be expressed in this so-called mesodermal plate, and surrounded by a ring of endodermal cells. And then everything else is ectoderm with a bit of specification of the aporal ectoderm. And the question is, how does that topology arise in, how does it set up in the first place from in a developmental mode? And how do we approach this? Well, we first looked at other bilaterians. For instance, this is a sea urchin late blastolar stage embryo as a cartoon, where we see that in the specification of these 3 germ layers, we have 3 different signaling pathways involved, like the Wnt signaling pathway, MAP kinase signaling, and notch signaling. So we thought, okay, let's have a look at these different signaling pathways and whether they also play a role in this specification. So, and for this you need a couple of marker genes marking these 3 germ layers. For instance, here snail would be, which is also in insects, marking the mesoderm, is here marking this, what we call the mesodermal plate, surrounded by brachyury in a ring here, and as well as FOXA, a typical marker of the endoderm. And then on the aporal side, we have markers which actually in bilaterians are typically anterior markers, actually forebrain markers like FOXQ2 and 636. This is the normal situation, but if we manipulate now, for instance, knock down the beta-catenin, so of the Wnt pathway, well, we get some striking phenotypes. So for instance, the mesoderm then takes over completely, so if you knock down, while all the others are gone. So that suggests that beta-catenin or BNP signaling, Wnt signaling is suppressing mesodermal fate, and if you take it away, then this mesodermal fate takes over the whole embryo. But if we also inhibit the MAP kinase pathway as a second pathway, then we see that we reduce completely the mesodermal plate. And by contrast, actually, this endodermal marker invades the territory of the mesoderm, while the aporal marker remains basically the ectoderm unaffected, as you compare with the control. So this would be, you would interpret this that, okay, MAP kinase is necessary to specify mesoderm, so in the middle, and if you take it away, then the territory is taken over by the endodermal markers. Interestingly, if you knock down both, beta-catenin and the MAP kinase signaling, then you see that actually both mesoderm and endodermal markers are gone, and now the aporal territory takes over completely. So which is like the default state of the embryo. So in other words, Wnt and MAP kinase define endoderm and mesoderm, and they push away this aporal ectoderm territory. So if both of them are gone, aporal ectoderm takes over. Okay, so this explains at first glance these segregation. When we do in a temporal aspect, we look at how these different territories are specified. We see that there's every hour of early development, you have a number of other transcription factors that are coming on in a cascade that are quite conserved, between cnidarians and the bilaterians on that level. So a lot of known factors that some people of you might recognize here, without going into detail. So these are all mesodermal markers here. And what you see by the temporal gradient here, that a little bit later, only at about 12 hours of development, you see some endodermal markers coming on. So the first thing that happens is you segregate only 2 different types of layers, mesoderm versus ectoderm. So the question is then, if you start with that, how is that endodermal ring of tissue surrounding the mesoderm actually initiated? this is again the question of signaling, and for this we had like a candidate signaling pathway which is would be suitable for making boundaries, and that's the notch signaling pathway, where actually notch and delta, I don't know what you whether you know them, but notch is a receptor for delta, the ligand, but both of them are membrane bound, unlike, for instance, Wnt and other signaling pathways where you have secreted signaling factors so that it's secreted out of the cell and can diffuse to a certain distance. While here in between notch and delta, the signaling only occurs between adjacent neighboring cells. Now, what we observe is that initially I start here with notch. Notch is expressed first early on everywhere in the whole ectoderm, and then you start by 10 hours or so, you start seeing some clearing here, which continues clearing, and this is the animal pole here. And at this point, you start seeing actually delta being expressed at this pole. So this is actually this mesodermal plate. And we do see that, so delta is expressed in the mesodermal plate, while notch is expressed everywhere else. So here also in the oral view, you see that this is this clearing of the mesodermal plate. But we want to know, of course, is there a cell boundary? So we did double stainings, double in situ hybridization, fluorescent ones, so that you see here again delta in red here and notch in green. And if we zoom in, we see that there's really a boundary from one cell to the other between delta expression domain and notch. And since you don't know from these pictures where is that boundary, is it between mesoderm and ectoderm or between endoderm and ectoderm? We used a FOXA as another marker for endoderm, and then double in situ with notch, we see that notch and delta overlap at this boundary. So we know that this boundary here is actually between mesoderm and ectoderm. Okay, that is just a correlation, but we can also manipulate this notch pathway, which you see here. Sorry, here. This is again delta, which is the ligand binding to notch. And when this happens, when a notch receptor is activated, it gets cleaved intracellularly by the gamma secretase, and that releases this notch intracellular domain, NICD, which then migrates to the nucleus where it binds to transcription factors, and together they activate targets of notch signaling in the cell where notch is expressed. And there is an inhibitor, or several, but we use mostly this so-called LY and this long number, LY, to block gamma secretase. So that blocks the notch pathway. And if you do that, and that's shown here, the upper row is again the control for, again, multiple markers, marking either mesoderm, endoderm, or ectoderm. And then you see only endodermal markers get downregulated in a dose-dependent manner of this notch inhibition, while the others are actually not affected at all. So neither mesoderm nor ectoderm. So what that means is if you need notch signaling to specify this endodermal ring surrounding you. This means, okay, it's necessary. In genetics, we often ask also the question, is it sufficient? So for this, we wish to overexpress a notch ectopically elsewhere in its activated form. And again, here the controls. And what we then did is we overexpressed just the NICD domain together with fused to M-cherry under promoter of TBP. And then you get patches of expression, ectopic mosaic patches. And then we looked for either mesodermal markers or endodermal markers. Mesodermal markers never are activated by this, but endodermal markers like brachyury and FOXA are ectopically activated by this patches of activated notch. In other words, notch is not only necessary, but is also sufficient to activate endodermal territory. And this would be explaining so that you first define this mesodermal plate, and then this expresses delta, while everywhere else is notch. And then at the boundary, it can start interacting with notch, so that you get at this boundary, you get actually delta notch signaling happening, defining this endodermal layer here. Okay, now coming back to the beginning, I mean, what we found now is that this interplay between these three pathways, beta-catenin suppressing mesoderm but activating endoderm, MAP kinase through ERG promoting mesoderm formation, and then the interaction here of notch delta providing actually the ring of endodermal markers. And there's a feedback loop between Wnt signaling and notch signaling that I didn't show you here in detail, but you can show that they actually cross-activate each other. And when we compare this with the situation again with the sea urchin, we do see that it's more or less exactly the same. And this is only focusing here on these signaling pathways, specifying these territories, but everything that happens downstream, this cascade of transcription factors that are continuing to specify the identity of this tissue, is to a large degree also conserved. And this is, again, as I mentioned, separated by about 600 million years. So conclusion one here is that this diploblastic cnidarian Nematostella is composed of three germ layer identities, which I consider homologous to ectoderm, endoderm, and mesoderm bilaterians. And these three germ layers are established by the same three signaling pathways as in the sea urchin. So when we think of mesoderm, we can also think of them not only as germ layers, but the name germ layer is because these are the earliest specification in embryonic development. So fate decision, so to say, from where normally then in particular tissues are derived from. So one of the key derivatives of the mesoderm in most animals are the muscles. And when we think of muscles, and that's really a hallmark of animals, because that they're moving, and because plants, no other organisms have muscles. I should say not all animals have muscles. There's almost all animals, but except for sponges and placozoans, so these very basally branching organisms. But they still have contractile cells that I don't want to go into. But what is really so remarkable about muscles is they allow us to of speed, for instance. And we think, for instance, of this cheetah here, which is of course very fast. But even the cnidarians that I work on, they're quite capable of moving themselves. So, for instance, this one is escaping this sea star by these coordinated movements. So the big question is, how do these different types of muscles actually evolved? And when here almost all of our muscles are actually of mesodermal origin, so which is a layer between ectoderm and endoderm, as you all experience, of course. While here we have only two cell layers in cnidarians. And we know a great deal about how muscles are made, in vertebrates, and how they are actually regulated by these neuromuscular systems. So by this interaction of sensory neurons, motor neurons, and muscles. And this interaction is super important. We know this also from diseases where, for instance, if the neurons detach from the muscles, like for instance in ALS disease, then if the motor neurons don't innervate anymore the muscle, then the muscle will just degrade, and get wasted. Now, but the situation in cnidarians is such that they mostly have so-called myoepithelial cells, where the cells are kind of a multifunctional thing. It's an epithelial cell at the apical end, even sometimes also with cilia. And at the base, they form these contractile, the myofibers, which elongate in one direction, which are, in close contact with this diffuse nervous system that could be either sensory neurons or, some other neurons. And also the regulation is not fully understood. Do they have also neuromuscular junctions like in vertebrates, or is it all diffusive signals that regulate them? And this is, to understand this, what is the relationship between these very different types of muscles is actually the key question, and so the co-regulation and co-evolution of these different populations of neurons is actually the subject of my ongoing ERC, but I don't have enough time to go into this, and it's also anyway a little bit preliminary, but I will tell you a little bit more about muscles in particular. Actually, when you type in muscle in say PubMed and ask how many entries you get over 1 million papers. And if you type in vertebrate muscle, you get still almost 1 million. So whereas if you type non-bilaterian muscle, which these guys belong to, plus other lower branching animals, you find 11. So there's a huge bias in the literature, and it's focused mostly on vertebrates, and to some degree also Drosophila, but really it's a huge bias in the literature. So I thought we should have to counter this. And this brings me to the situation again for muscles and with this phylogenetic tree. So again, here's the sister relationship. The cnidarians, they fall into different classes that I will not go into in detail here. Nematostella belongs to the anthozoans, which are here, so to which also corals belong to. But the situation is mostly the same, but they have these myoepithelial cells. Here, this is from Hydra, where you see these longitudinal fibers at the base of the epithelial cells, both in the ectoderm as well as in the endoderm, where they're a bit shorter. And while in bilaterians, they mostly arise from the mesoderm layer, and then they come in vertebrates as well, at least, but in most other insects, most other bilaterians as well, in 3 different flavors. So you can either have them as striated muscles or as smooth muscles. But among the striated muscles, there is the fast contracting striated muscles of our here, for instance, here, the skeletal muscle. They are striated and fast, while the cardiac muscles are also striated, but they're not so fast in their contraction, and also the regulation is totally different. Well, we cannot voluntarily control our heart. It's autonomously regulated. And then there are the smooth muscles, which are, for instance, mostly lining our gut and are responsible for the peristalsis in our gut system. And they're considered to be slow. And interestingly, they're marked by, in their development, by different transcription factors. I just mentioned here one, for instance, GATA 456 for the slow, whereas this BHLH transcription factor MyoD, a very famous transcription factor regulating the differentiation. So if you transform a fibroblast with MyoD, it transforms into a striated muscle. So it's a master regulator of this whole cell type. Now, okay, there is, in terms of evolution of these kind of situations, this scheme has been put forward first by George McKee in 1970, then revived by Detlef Arendt, where the idea behind this is that cell type evolution and diversification might occur by some sort of subfunctionalization and segregation of labor. So if you had first an ancestral cell type which unites everything, it's an epithelial cell, it's a sensory cell, and it's a muscle cell all in one. And then step by step, you get then you segregate these different parts. First, you separate it by a muscle, then you make one sensory plus motor neuron, and then you divide the neurons into sensory and motor neurons. This is a nice conceptual idea, but it's, and people thought, okay, the cnidarians, actually, they have these, and they're kind of, then this is the way it went in the other lineages. But of course, it's kind of an ad hoc scenario. And I try sometimes to compare this with these kind of paintings where, well, if you start at one corner here with birds and then the other end, Now I swapped into this wrong corner. Oh, so it's the wrong direction. Oops, can only go forward. So here we are. So yeah. So as I said, I mean, I think it's a nice scenario for thinking about this problem, but in a way you can convert everything. Where's my pointer? Not visible. Oh, over there. Dangerous. Here the birds, and by steps you can convert everything into, say, even a play chess. Yeah. So if you wish. Yeah. So it doesn't mean that really evolution works like this, right? You know this better than me. So I think we need better, we just have to look at the data, and then we can come up with, based on the data, we have to look at the scenarios that we can develop from there. And how I look into this is from a cell type level, and conceptually, there I try to decompose the cell type concept into three parts, which is the first part is really the developmental part, how this cell type is made, and the key factors here are the so-called terminal selector genes, which are transcription factors, and often the synonym is a co-regulatory complex. So whether they form a complex or not, or whether they just act in parallel, is a bit of a question. But there's a number of transcription factors that are expressed in even in the differentiated cell types, and they're there to maintain that particular cell type. Yeah. And this combination of transcription factors differs between the different cell types. That's this idea, and they regulate the phenotypic level of this cell type, which is either structural protein genes like myosins in the case of muscles, or synaptic scaffolding proteins for neurons, for instance. But also, of course, other phenotypic traits of that cell type, which are not about the shape, but actually more their function. So for instance, physiology, like ion channels and neurotransmitters, which differs between different types of neurons and also different muscles. And you will see why I emphasize this differentiation, because I think we have to sometimes look into these different levels of gene expression when we talk about cell types. And then we can ask, whether given cell types between organisms are homologous, or whether they have actually convergent evolved. Interestingly, if we look only into vertebrates, and this is work that was also done by Detlef Arendt and Thibault Brunet in a review, when they looked only at transcription factors and there's this signature between these three different types of muscles, what they found to somewhat surprising at first is that the two striated muscles, so the skeletal and the cardiac muscle, actually are not so similar at all in the setup of transcription factors. While the smooth muscle myocyte and the cardiac myocyte, they share a great deal of these transcription factors, suggesting that they may actually have also ontogenetically the same origin. And indeed, I mean, the heart is a specialization of a blood vessel, and the blood vessels are surrounded by smooth muscles. So after all, maybe it's not so surprising, but it illustrates that, what we see phenotypically, say, for instance, here, striation here and there, does not automatically mean that they are closely related. Okay. So how's the situation again here in cnidarians? We looked in Nemapstella again. I will show you that they have 4 different types of muscles. They're all smooth muscles in the polyp, and they occur at different locations. So if one is in the tentacle, I will show you pictures in a moment. There's retractor muscles, which are longitudinal along the length of the tentacles in the ectoderm. And then the inner layer, we have the circular muscles. We have also the circular muscle, which is a continuum here in the whole body column. So all the inner layer has a circular muscle going around this. If we make a cross section here in green, CM circular muscle, and in a cross section you find that these sea anemones, they have 8 mesenteries, which are like foldings going in and out into the gastric cavity. And this is one of those folds that you see here, which from the body wall goes into the gastric cavity and then goes back in and then unites back. So it's always diploblastic, but what you see then in the middle of it, on one side, they have the longitudinal mesentery retractor muscle, and at the base of them, they have this parietal muscle. So I take this example as an example of how could in evolution the diversification of different cell types actually occur. So first, some nice pictures. So again, these by phalloiding stainings, and we have also transgenic lines for this. These are the longitudinal muscles of the parietal muscle. In a cross section, you see them here, and this strong retractor muscle in the mesenteries. And they're called retractor muscle because if they contract longitudinally, they pull in all the tentacles and the pharynx. So it's a reaction that they need for escape, as an escape reaction. And where here you see these circular muscles, which are thinner, which are responsible for the peristalsis. But here, these muscles that we see here in green, what is highlighted in green is phalloidin staining for the F-actin part, so which is only the basal part of the cell. Indeed, when you do electron microscopy, it's a bit difficult to see maybe, but the muscle part which you see in green is only here at the very base, attaching to the extracellular matrix, but you can follow that cell to the very tip, and there it is anchored to other epithelial cells. So it's still an myoepithelial cell with a basal, longitudinally elongated contractile element. And in the tentacles, it's similar, but they are actually ectodermal, as I mentioned. Now, we did single-cell transcriptomics and focused on the muscles. And what we found is that these 2 retractor muscles in the tentacles, so here TR, tentacle retractor muscle, and MR is the mesentery retractor muscle, the strongest one. They are actually sharing the expression of quite a number of these structural protein genes, muscle protein genes, while these other 2, parietal muscle and circular muscle, they're basically expressing a whole other set of these structural protein genes. This was somewhat surprising because if in the ultrastructure, for instance, this CM and PM and MR, they are actually they look like different flavors of the same thing. So I would have guessed that this is actually they express the same genes. This was validated by in situ hybridization, so that these genes are indeed specifically expressed in only these longitudinally oriented muscles, while these guys here are expressed more broadly in the circular muscles and elsewhere. So if you can also regroup these genes by gene families, and for instance, then we have, for instance, only tropomyosins and its paralogs, because all these genes occur in multiple paralogs. And then we notice that if they have multiple paralogs, then in many cases, each of these paralogs segregates to either one of the 2, so of these tentacle retractor muscles, one of those guys, or on the other side. There's a few which are expressed more broadly and ubiquitously, but in most cases it's a segregation, so subfunctionalization of these paralogs into different types of muscles. Now, what's interesting is that why do they do that? Or of course we don't know, but what unites tentacle and mesentery retractor muscle is their contraction speed. They are we measured the contraction speed, and they're actually 50 times faster than, say, for instance, the circular muscles, which you see here, which are responsible for the body peristalsis. So we don't know, of course, and we just believe that these different structural protein genes, which are paralogs of these guys, are conveying this higher physiological property of being faster or slower. Yeah. So of course we cannot nail it down to a single one. It could be the combination of all of them. But obviously so many gene duplications, which were then subsequently segregating by subfunctionalization and then led to the emergence of these different properties also of different muscles. Even though they're all smooth, we have fast contracting and slow contracting muscles. Now, I think, is that? Yeah. Here you see the contraction, for instance, of the retractor muscles in this little movie. And here, this illustrates in a transgenic line the peristalsis movement that happens in of course, in a time-lapse movie. So that happens a little bit like in our gut system as well. So constant peristalsis. Good. So then we wondered, how are these two different major classes of fast and slow muscles specified by transcription factors? And first we looked at transcription factors that are involved in muscle formation in bilaterians that are listed up here. And if we have them at all, for instance, MyoD, the famous MyoD doesn't exist in the genome. Yeah? In non-bilaterians, you don't find it. So that's bilaterian specific. But others you find, but they are mostly expressed in all kinds of other cell types. Sometimes they are also expressed in muscles, but definitely not specifically. So you find them also even more strongly expressed elsewhere. But then we looked in a more unbiased approach what is expressed specifically in those cells. And to our surprise, if we focus here now on these two fast contracting muscles, which share the same set of structural protein genes, as I just showed you, then to our surprise is that they don't share the same set of transcription factors that seem to govern and regulate this phenotype. And if we focus on a few more members of those here, for instance, there's the BHLH protein, E protein, which is expressed very broadly because that's a common factor that you need for any BHLH factor. So it's not a surprise that it's expressed broadly, but it's strongly expressed in the tentacle retractor muscle. And I will go into one more detail in a moment. And here's another BHLH factor, NEM64, that I will spend some more time on it. And flanked by its paralogs, NEM24 and NEM7, which are expressed in the other muscles. So they seem to be largely exclusive to each other. And another BHLH, HAN2, which is not expressed in the fast muscles, but only in the slow muscles here. Okay, so I want to focus on these two factors first, these two BHLH proteins, for which, just as a reminder, BHLH proteins, the basic helix transcription factors, and they always form dimers, of which one of them is invariably the E protein. For instance, here in the example with MyoD, then it binds with a specific factor. So it is often ubiquitously expressed, and then in each cell type, it can bind to various more specifically expressed BHLH proteins, and together then they have the function that actually is conveyed mostly by this player here. So if we have made a knockout with CRISPR of MyoD, here you see the control, just the muscle fibers and gene expression by myosin heavy chain. If they first develop normally until they start making the muscles, and then you see it's completely messed up, and then they die. And they actually also don't express these structural protein genes. So, but of course, maybe not so surprising because that is expressed in all muscles, but also in other cell types. But then we looked into this very specific VHLH protein, which is only expressed in this tentacle retractor muscle. Here you see some in situ of individual cells at the base. And for this one, we also did a CRISPR mutant. And at first we thought, okay, there's no phenotype because the polyps looked at first glance normal. But then we noticed actually that the tentacles are basically hanging, and they cannot they cannot move them really, because when we stained them, we noticed here's the control. These are these tentacle retractor muscle, longitudinal, and they're completely absent in this mutant. And this was confirmed also by overlaying, doing single cell transcriptomics. And you see here, the black dots are the mutant cells in the muscles, and what is missing is any cells from the tentacle retractor muscle in the mutant. Yeah, and if you do also in the mutant, if you look at structural protein genes compared to the wild type, which are, because they share these structural protein genes between the tentacle retractor muscle and the mesentery, you still see them expressed in the mesentery retractor muscle, but they're completely absent in the tentacle. So we think that this NEM64 is somewhat like a master regulator of this tentacle retractor muscle. Absolutely necessary, and if you don't have it, the whole cell type is gone, but only that cell type. And that cell type is something special because, as I briefly mentioned before, and this is a transgenic line here, is that this is the only ectodermal muscle that we have in this organism. All the others are from the inner layer. And they're born here at the early stage, from an epithelial cell. And unlike the other myoepithelial cells, which are always attached as epithelial cells at the base, at the apical end, these ones actually detach, and then they take on a position which is basically at the base of the epithelium, like neurons do. Actually, when a neuron is born, they also do the same, and then they extend their myofibers longitudinally. So it's quite interesting, and there's another peculiarity about this particular muscle in the tentacle, is that it's co-expressing a number of transcription factors and other neuronal markers, so which are really specific for neurons, which you don't find anywhere in the other muscles. So one of the key factors here is SOXB2, which is a transcription factor that was previously shown also functionally. If you knock down SOXB2, then you basically lose almost all neurons in a knockdown. And it's interesting that this is co-expressed in the same cells, together also with these muscle markers. And this extends to other markers, where, for instance, here we have in the single cell transcriptomics, so here this is a tentacle retractor muscle next to the other muscles. And here we looked at different postsynaptic proteins, but also acetylcholine receptors, I-glutamate receptors, GABA receptors. And what you might notice is it's expressing quite a number of different receptors. Almost none of them are expressed in the other muscles, but many of them are shared with different kinds of cnidocytes and neurons. So again, makes this like phenotypically almost like a hybrid cell type between a muscle and a neuron. Yeah, very peculiar. We don't know yet. What we don't know is whether they can also signal like a neuron, like whether they have actually, this is something I would really love to see, whether they can only receive signals like a neuron or also send out signals. So what is interesting here, if we now, and that's why I come back to this comparison between the regulatory profile and the structural profile of the transcriptome fingerprint. If we do, say, like a little tree of the regulatory profile, then we do see that the tentacle retractor muscle of the fast one and the mesentery retractor muscle are separated. They have actually very different transcription factor profile. Whereas actually, if you look at the structural protein genes, they would be united. And if you do everything all in one bag, then since the transcription factors are the more rare genes of this, it's mostly driven by by the structural protein genes. So then you would assume, okay, it's a sister cell type, but actually it's not. So I think it's worth actually always looking separately into these different things. So here's our scenario, what might have happened. First, this is more in a ontogenetically, or a mixture of both. So E protein is shared with all lineages, ectodermal and endodermal. And then you had here diversification of some transcription factors like this BHLH hand and NEM7 and 24, as well as NEM64. And what we think is happening is here that these structural protein genes, they have duplicated, all of them independently of each other, and then were recruited, step by step into these different, conveying the different physiologies. But there was another gene duplication of these transcription factors, or actually two, which gave rise to NEM64 in the tentacle, being expressed in the tentacle. But since it's so similar, it was still driving the same program. So basically co-opting this structured protein program into the tentacle retractor muscle. Yeah, another aspect that I didn't talk much about, but this the transcription factor profile of this parietal muscle unites hand, TBX110, TBX20, NKX2.2, and these are actually factors that we do find in the bilaterian cardiac muscle, so which is the only one which is really conserved among the muscles, also in bilaterians. So that's quite interesting. One more step, and then I'm almost done. So this NEM64 gene is here, this in yellow. When we do the gene phylogenies, we do see of all these BHLH proteins, first of all, it belongs to a larger group of BHLH genes which unite hand, twist, paraxis, which are in bilaterians indeed muscle-related transcription factors. And Matostella has actually homologs of each of them. But then the cnidarians went a radiation here to expand on some of these other factors. Actually, I should be more specific. It's a sea anemone specific radiation. And that gave rise to these four paralogs, of which one of them is NEM64 only being expressed in the tentacles. So, and indeed, we do find, and this is the phylogenetic relationship between them, they're still on one chromosome, and next to actually twist and paraxis, or others of the same family. But most of them are expressed in the mesoderm here, but only this one, NEM64, is expressed in the ectoderm and driving this program of a hybrid cell type. Very weird, yeah. Whereas they share all this NEM6027 and these other guys, they share kind of the same regulatory profile. So here's our scenario, what might have happened. So coming from a first ancestor gene, which duplicated twice, and then there was translocation. So two of them stayed in tandem, NEM7 and NEM24, and they were kind of copying their cis-regulatory elements, which would drive them into, into the mesenteric retractor muscle. And this is a fast muscle proteins. NEM64 was translocating to a different location where I postulated, okay, there might be already by chance some regulatory elements that it was capturing or hijacking, which drives it actually into the ectoderm, and also taking input from neuronal determinants, and which is then the reason why it also co-expresses SOXP2 as a parallel program between the muscle program. Yeah, and this was now recently by the group of Arnau Sebel-Pedros, who does a lot of genomics. He looked into this by ATAC-seq and looked into how we can define the cell types based either on gene expression profiles or on the basis of their cis-regulatory elements identified by single cell attack-seq. And what you do, if you, for instance, if you take whole gene expression profiles, then you would actually unite the muscle transcription, the tentacle retractor muscle and the mesenteric retractor muscle together, as I mentioned before, because it's driven by the same set of structural protein genes. But if we look at attack-seq peaks and the variable peaks, then we do a tree based on this, then they actually separate on very distant way levels of this tree. And if they analyzed actually the underlying regulatory sequences underneath these peaks, then even then also they're getting separated again. And you can also analyze this in such a way that you look at the accessibility for transcription factors in the cis regulatory regions of these two muscles, the mesenteric retractor muscle and the tentacle, and you see that actually they separate completely, and you find here quite a number of factors that are known for neuronal development as drivers of neuronal development, whereas here we find many conserved markers of muscle development. Zooming into the locus of NEM64 and its paralogs, you see here NEM64, and here it's two paralogs, NEM24 and NEM7. Then you see that here these are the conceived regulatory signatures in the CRMs of these genes, which are completely distinct from the ones that actually have been acquired by NEM64, where actually you see again also SOXB2, this key, and SOXC2 key regulators of neuronal development. Yeah, so which confirms actually what we postulated, that it jumped to a different region, acquired a completely new input, which drives it into not only a new region where it co-opted the structural protein genes, but also acquired new features by this. I will skip that for time reasons, but we looked into striated muscles now in jellyfish, but happy to discuss that briefly afterwards. But would like to start with this one. So just as this is a totally speculative and open ending. So we know, of course, there are species trees that we can generate based on whole, nuclear genes and so on. And Arndt von Haesler has been instrumental to build algorithms for this. And you're familiar with that. The same principles have been applied to cell type trees based on extend cell types and their profiles. But I think if you want to understand also the processes, now I come back to the processes, how we get there, it actually, I think it's worth looking into some more details, what's happening on the genomic level and individuals. And I think taking this scheme as an analogy from bacteria, eukaryotes, and archaea, where there's a lot of horizontal gene transfer, which actually confuses at some point, how these different lineages were actually resolving into individual separated lineages. And of course, there's all these events that happening that can actually lead to also something like hybrid cell types that, if they make sense, then they may be selected and give rise to actually a diversification of cell types as well. But I think this is one out of several mechanisms that might be at work in order to give rise to more or less complex organisms. And the complexity of these organisms, as a large approximation, probably can be measured by the numbers and diversity of different cell types. With this, I would like to stop and thank my group, and the people highlighted here in bold are the people who have made a major contribution to this story, and here are the funding bodies. Thank you.